Intermediary metabolism is essentially what individual cells are going to be doing with the nutrients that they acquire, and what they can do with them, what they do choose, so to speak, to do with those nutrients. So it's metabolism at a cellular level. We're also going to be talking about metabolism at an organismal level a little bit later. So any nutrients that are absorbed at the intestine are going to travel first to the liver via the bloodstream for repackaging and distribution. You can essentially think of the liver as being like a distribution warehouse for Walmart, for example. You don't have individual vendors sending trucks to all of the different Walmarts throughout the world. Individual vendors like Coke or Pepsi or what have you, any particular vendor is going to distribute first to a Walmart warehouse, and then that warehouse is going to distribute to the individual Walmart stores. So all of the material is going to go to the warehouse first, and then from there it's going to go to individual stores. In the case of nutrients, all of the nutrients go to the liver first. The liver is the warehouse, and that's where those nutrients will then be repackaged and distributed to the tissues that actually need the materials. So there's lots of different types of nutrients, of course. We're going to talk about intermediary metabolism in the context of the three major components of the three major macronutrients. So proteins, lipids, and carbohydrates are macronutrients. Proteins, of course, their constituents are amino acids. Lipids, their constituents are fatty acids. Carbohydrates, their constituents are glucose and other free fatty acids. From amino acids, you can, of course, produce energy. You can catabolize amino acids to form energy to drive the cellular processes of the cell. You can also take amino acids and utilize them for protein synthesis or glycogen synthesis. In the case of fatty acids, of course, you can utilize fatty acids for energy production as well. But you can also use these components to form new lipids or other compounds like eicosanoids. Certain types of fatty acids can be utilized for hormone synthesis, for example. So you can use those fatty acids for structural uses like forming new lipids or new cell membranes, or you can utilize it for the production of other important products. In the case of carbohydrates and their constituent sugars like glucose, their function is largely energy production. So you can produce energy from them, certainly. About the only other thing that you can do with glucose or other simple sugars is to store them as glycogen. That stored glycogen can then be utilized as an energy source later on. So glucose, carbohydrates can really only be used as an energy source either immediately or later on, whereas fatty acids and amino acids can be utilized for energy production now or later, or they could be utilized for other structural uses because they are the building blocks of so many important molecules and structures within the body. So the choice, so to speak, of a cell as far as what they do with these different nutrients is going to depend upon hormonal influence. So we talked about how a starving fish will do different things with nutrients than a fish that has been consuming food regularly. The way that those cells are influenced and the way that they know the general state of the fish, whether it's starving or well-fed, is through the endocrine system and through hormonal influence. We'll talk a little bit more about the endocrine system later on in this course, but for now we're going to talk about it in a very simplistic sense. So what I'm showing you here is glucose metabolism by a hepatocyte or a liver cell. The process could be the same for any number of other different types of cells of the body. We're just going to deal with the liver because of its key importance in nutrient trafficking and distribution. So the hepatocyte may be absorbing a molecule of glucose that was delivered to it via the bloodstream. Through the action of an enzyme called glucokinase, that glucose molecule is then transformed into something called glucose 6-phosphate. This is the point at which hormonal influence can make the decision, so to speak, whether that glucose 6-phosphate is utilized for catabolic processes or anabolic processes. So glucose 6-phosphate can be broken down further to 3-carbon intermediates formed into pyruvate, enter the Krebs cycle, and be utilized to produce NADH and ultimately ATP. This is the catabolic side of things. This is what you will do with a glucose molecule if you needed to produce energy. Catabolic means breaking down largely for energy purposes. So when we talk about any kind of catabolic process, we're breaking down a structure for energy production. That's one option. On the other hand, if the fish is under a different hormonal influence, or rather if the hepatocyte is under a different hormonal influence, that same glucose 6-phosphate could be transformed to glucose 1-phosphate, and then through the action of an enzyme called glycogen synthetase, be formed into glycogen. This is the anabolic option. This is so anabolic. You probably heard the word used with reference to anabolic steroids that are used by bodybuilders to bulk up. Anabolic processes are those that are not about breaking down. They're the opposite. They're about building up. So in this case, we're utilizing glucose to build up our glycogen stores for energy production later on. So we're storing it for use later on. So again, this is the option. We can take glucose and we can transform it into energy through catabolic processes. We can transform it into glycogen through anabolic processes. And the determination as far as whether that glucose molecule enters the catabolic pathway or the anabolic pathway is going to be driven by the hormonal state of the fish. We'll talk a little bit more about how that works later on. Now, what about amino acid metabolism? Here we have the same hepatocyte, but now it's going to be absorbing an amino acid. Again, something that would have been delivered to it via the bloodstream following absorption. So first of all, that amino acid could be used for the synthesis of new protein. So it may be taken up directly by the hepatocyte's ribosome, one of its ribosomes, and a new protein could be synthesized. So it's a new protein, we're building something up, that's an anabolic process. If, on the other hand, the ribosome doesn't need that particular amino acid, we can then break that amino acid down into its carbon skeleton, forming those 3 carbon intermediates. In fact, the very same or very similar types of 3 carbon intermediates to what we saw on the previous slide for glucose. So this is that process that we discussed before, where we take, through the process of transamination, we transaminate something, any amino acid to glutamate, and then we can deaminate that glutamic acid or glutamate so that we have the ammonia or the amine group removed and then only the carbon skeleton left over. This is that exact process. So the amino acid can be transaminated or deaminated as needed to form those 3 carbon intermediates if it's not needed for protein synthesis. Those 3 carbon intermediates, of course, can enter into that same catabolic pathway that we saw before. They can be transformed into pyruvate, enter the Krebs cycle for NADH and ATP production. That's the catabolic option. So, of course, we have to go through the trans and or deamination process, but that's one way that we can produce energy, or that's the way that we can produce energy from that amino acid. There, of course, is a second anabolic option for those 3 carbon intermediates. Rather than going into the catabolic pathway, they could go into the anabolic pathway and be utilized to produce glycogen in the exact same way that we saw for glucose on the previous slide. So in the case of amino acids, we've got 2 different anabolic options. We can form glycogen for energy storage, we can form new proteins, or we can enter the amino acid into the catabolic pathway for energy production immediately. And again, how this particular process works and what pathway that amino acid takes is going to be influenced by what amino acid is and whether it's needed for protein synthesis, and just as importantly, the hormonal influence and the overall anabolic or catabolic state of the organism. Now, with respect to free fatty acids, you can see that this slide looks awfully similar to the protein one. So a free fatty acid can be absorbed by the hepatocyte, and it can be immediately used for new lipid synthesis. That's one anabolic option. Through a process called beta oxidation, that free fatty acid can be formed into, you guessed it, 3 carbon intermediates that can enter into the Krebs cycle for energy production. Alternatively, those same 3 carbon intermediates could theoretically be used to produce glycogen. Now, generally speaking, most fatty acids that are consumed that are not needed immediately for new lipid synthesis for structural reasons, they may still enter that new lipid synthesis anabolic pathway anyway, because you can store free fatty acids as lipid. You can store energy that way. You don't have to transform them into glycogen to store them. In the case of amino acids, if there's no need for them for structural reasons, if you're not going to synthesize protein from them, you have to transform them into glycogen. You can't just store free amino acids. Now, in the case of free fatty acids, you can do that. You can produce new lipid for a reason, for new cells, for new cell membranes, for eicosanoids and other products, or you can just store it as triglycerides as fat. So you don't necessarily have to go through that process to produce glycogen, although you could. So again, the choice between the anabolic processes and the catabolic processes are going to be driven by hormonal influence. You can see that there's a lot of similarity between these intermediary metabolism pathways. In the case of catabolic processes, they're almost identical. In the case of anabolic processes leading to glycogen, they're also virtually identical. But again, keep in mind that glycogen synthesis, although theoretically possible from lipid, generally speaking doesn't happen because lipid can be stored essentially as is, unlike carbohydrates or glucose and simple sugars and free amino acids. I also want to note that that new lipid synthesis that we talk about and utilization of free fatty acids doesn't necessarily have to be exclusively related to the formation of a new triglyceride molecule or a new phospholipid molecule. It can include biotransformation of existing fatty acids to form new fatty acids. So on the left-hand side of this figure, you can see 18,3N3, 18,2N6, and 18,1N9. These are three different 18-carbon fatty acids that that hepatocyte on the previous slide could have absorbed. Through a series of elongations that add additional carbon molecules or desaturations that add additional double bonds, those 18-carbon fatty acids on the left-hand side can be transformed into new 18-carbon fatty acids like 18,4N3, 18,3N6, or 18,2N9, and then they can be transformed into even longer chain ones like the 20-carbon and 22-carbon fatty acids. Those of you who are aware of fatty acids or work in my lab and do a lot of lipid biochemistry work, you'll know that certain of these fatty acids, specifically 22,6N3, 24N6, and 25N3, these are really important fatty acids, the long-chain polyunsaturated fatty acids that have a myriad different physiological roles in functioning of the nervous system, vision, reproduction, heart health, etc. So just keep in mind that when we talk about new lipid synthesis by the cells and intermediary metabolism, we're not just talking about taking whatever fatty acids come in and forming new triglycerides from them. We can also be talking about taking fatty acids that are absorbed and transforming them, depending upon the species. We're going to start getting into some of the hormonal or endocrine aspects of digestion and metabolism now. And so I just want to put this slide up again as a reminder. So we have different types of hormones that are involved in this process. Some of them we've already talked about. We've talked about some of the digestive hormones that are involved in inducing different components of the digestive process. We're going to review that in just a moment. But we also have metabolic hormones. These are the ones that are going to help the cells make the decision, so to speak, between whether they engage in anabolic utilization of resources or catabolic utilization. So keep in mind again that virtually all of the processes related to food acquisition, digestion, and utilization are going to be under endocrine control or under the influence of hormones. So the feeling of hunger or feeding behavior is driven by hormonal processes. The release of digestive enzymes, the churning and contraction of the stomach in fishes that have a stomach, those are all influenced by hormones. Certainly, anabolic or catabolic utilization of resources is going to be influenced by hormonal state. And so we're going to talk about the digestive and metabolic hormones. Not all of them, just a handful of them that I want you to be familiar with. The digestive hormones we've already talked about a bit, but we're going to review them before we get into the metabolic hormones. So these are, this table over the next slide as well includes the hormones, some of the hormones of the gastrointestinal system of fish. We've already talked about some of these. So gastrin is one of the digestive hormones. It's produced by the stomach, it acts on the stomach, and it functions to induce the release of hydrochloric acid and pepsinogen, as we've already discussed. Cholecystokinin, this is another digestive hormone. It's released by the anterior intestine in response to the increasing availability of those free amino acids, free fatty acids. It acts upon the gallbladder primarily, as well as the pancreas and the brain, and it's going to stimulate contraction of the gallbladder, release of bile into the intestine for emulsification of lipids that may be present there. It's also going to act upon the pancreas to induce it to release additional types of enzymes for digestion. It's also going to decrease feeding behavior. This should kind of make sense. If you're already well into the process of digesting and absorbing an existing meal, existing food that's in your gut, you probably don't need to be out there acquiring additional food. And so cholecystokinin will help to suppress feeding behavior and allow the fish and its systems to focus on utilization of what it's currently got in its gut. Some additional hormones are listed on this table. You saw bombesin on the previous slide. You can see some additional information here about several others like vasoactive intestinal peptide, somatostatin, neuropeptide Y-like peptides. These are other aspects, other hormones of the gastrointestinal system that I don't necessarily want you to know about, or I won't be asking you questions about for the exam, but they're certainly very important in the context of digestion and utilization of nutrients. And so if you're very interested in this, there's some additional references here as well. At the very bottom here, you see secretin. This is another of those digestive hormones that we have already talked about. This is one that is released again by the anterior intestine. It acts upon the pancreas and induces the release of bicarbonate ions as well as various digestive enzymes from the pancreas. So with respect to the digestive hormones, the ones that I want you to know about are secretin, cholecystokinin, and gastrin. I think those are the most important in terms of understanding the generalized digestive process, but please understand that there are many, many more hormones that are involved in the digestive process as well. So even though I'm not going to be asking you specific questions about that, just be aware that the system is a lot more complex than just those 3 digestive hormones. Of course, it's equally complicated when we talk about the metabolic hormones. There are lots of different hormones that are involved in regulating whether cells of the body engage in anabolic or catabolic processes. But we're going to talk about 2 primary ones, one of which is likely already familiar to you, and that's insulin. Insulin is considered to be a largely anabolic hormone. It's produced by the pancreatic cells and causes the cells of the liver, but also all the cells of the body, to uptake glucose and other resources for anabolic utilization. The counterpart to insulin is glucagon. Glucagon is considered a catabolic hormone. It's also produced by the pancreatic tissues, different cells within the pancreatic tissues, however, and it acts upon the liver as well as other sources or other tissues in the body, other cells in the body, and induces them to break down available resources to liberate energy, liberate glucose and other resources for energy production. This figure is showing how these 2 particular hormones function together with respect to regulating blood sugar levels. Now, of course, blood sugar levels are very tightly regulated in humans and other higher vertebrates. They are regulated in fish as well, although to a less strict extent. But it's important to maintain proper levels of blood glucose, blood sugar levels in all organisms, and it's these 2 hormones that interact to ensure that proper balance. So if blood sugar is rising, for example, following a meal, and you have increasing availability of nutrients being absorbed by the intestine, distributing them to the liver via the bloodstream, you would have an increase in blood sugar as well as other nutrients available in the bloodstream. In response to that rise in blood sugar and availability of other nutrients within the bloodstream, the pancreatic beta cells would release insulin to tell the rest of the tissues of the body, hey, we've got a positive flux of nutrients here, let's utilize these for anabolic processes. On the other hand, if the levels of blood sugar and other nutrients drop very low, that's going to be an indication that there aren't enough resources for energy production, and energy production is going to be limiting. That's then going to induce the pancreatic cells, the alpha cells, to secrete glucagon. Glucagon will tell the cells of the body then that, hey, we are in a negative nutrient flux, we don't have resources coming in, we know that we have energetic demands. In order to meet those energetic demands, we're going to have to break down the resources that we have. So the liver will begin to break down glycogen, Other cells of the body will break down other stores that they have available of either glycogen or lipids, or in some cases even proteins that are present, breaking them down into those 3-carbon intermediates so they can be funneled into the Krebs cycle for energy production, so the ATP is not limiting for various cellular processes. So again, this is just an example of how two particular metabolic hormones, insulin and glucagon, balance each other and help the organism make the right decisions, quote unquote, to ensure proper utilization of energetic resources. So here's a figure to illustrate the functions of insulin and how it's going to affect 3 different types of cells: liver cells, adipose or fat cells, and muscle cells. So we talked about how insulin is generally anabolic, so it's going to encourage the cells to build up different resources, or the cells themselves. So you can see that it has anabolic effects on all of these cells. So in the case of the liver cell, it's going to encourage those liver cells to absorb glucose from the bloodstream. Again, remember that insulin's function in part is to regulate those blood levels of glucose and ensure that they stay within a tight range, a relatively tight range in the case of fish. So the liver cells are going to be absorbing glucose, and those glucose molecules are, in the case of insulin influence, are going to be utilized to produce glycogen. That's what's shown there in pink. So we're taking the green glucose molecules, we're transforming them to glucose 6-phosphate, and then we're transforming those molecules through glycogen synthetase into glycogen for storage. That's primarily what the liver is going to do with that glucose that it's been told to absorb. So that previous slide that we showed of the hepatocyte, where we had the arrow indicating hormonal influence, this is that hormonal influence. Insulin is directing those cells to absorb the glucose and then form glycogen from them. In the case of the adipose cell, insulin is also encouraging those fat cells to absorb glucose, not for the production of glycogen, but rather to absorb glucose to produce energy and to form a glycerol for the synthesis of new lipids. So it's going to be absorbing glucose, it's going to be forming glycerol, it's also going to be absorbing free fatty acids under the influence of insulin for the synthesis of new lipids, either for storage purposes or for structural aspects. So we could be producing more triglycerides, we could be producing more phospholipids for more cells, etc. But again, these are generally anabolic processes. In the case of the muscle cell, insulin, of course, is going to be inducing the absorption of the uptake of glucose for this cell as well, but it's utilizing glucose in this case not for glycogen synthesis or for glycerol production, but rather for the production of energy. That energy is going to be utilized by these muscle cells to fuel the process of synthesizing new proteins. Insulin is also going to encourage or induce the uptake of amino acids by muscle cells as well, so that that free amino acid pool is full and then can be utilized to synthesize those new proteins. So in each of these 3 cases, insulin is having an anabolic effect. It's encouraging the uptake of available resources. That's going to help with balancing blood levels of those nutrients, and it's also going to encourage those cells to engage in the anabolic pathways that are available to them for glycogen synthesis, new lipid synthesis, or protein synthesis. In the case of glucagon, the catabolic counterpart to insulin, many of these processes would be reversed, where you'd be taking the same cells and proteins that are available, glycogen that's available, fat that's available, would be broken down to liberate energy and form ATP to ensure that those energy needs of the cells and of the organism are going to continue to be met. This is a rather complicated figure, but if you understand everything that's on this, then you're going to understand the basic digestive process and the hormones that are involved in that. So we're going to step through this fairly slowly, just so that folks can hopefully make sense of it. I hope it will be a useful study guide. So we're going to start with that picture of that steak there. We have a food item that's being consumed. The food item is consumed by a fish. That food item is going to distend the stomach and increase its size. That's going to cause a nervous impulse that will then induce the G cells of the stomach cells themselves, to produce gastrin. That's our first digestive hormone. The gastrin itself is going to work on the parietal cells, or the oxynticopeptic cells, to produce hydrochloric acid. The hydrochloric acid and the gastrin are both going to function to induce the release of pepsin from chief cells. Now, throughout this presentation, you've heard me say things like oxynticopeptic cells. You've also seen parietal cells and chief cells shown on figures like this one. In some animals, it's likely that there are separate cells that produce hydrochloric acid and cells that produce pepsin. So parietal cells are producing hydrochloric acid. Pepsin is being produced by the chief cells. In the case of fishes, it may be that those cells are the same, these oxynticopeptic cells. So it may be the same exact cell producing both. In this case, we're showing it as being separate. But understand that when we're talking about oxynticopeptic cells or parietal cells and chief cells, what we're talking about are cells that are producing hydrochloric acid and or pepsin in response to gastrin. Okay, so then we have hydrochloric acid being released, and then we have pepsin. So pepsin, as is shown on the top of this figure, we have pepsin that's going to be attacking proteins as an endopeptidase. It's going to be snipping those proteins in the middle and producing smaller peptides. Once those peptides make their way into the intestine, they are going to have subsequent functions later on. We'll talk about that in just a moment. In the case of the hydrochloric acid at the bottom of the figure, once that hydrochloric acid and the acidified digesta reach the anterior intestine, that's going to induce the S cells of the anterior intestine to produce secretin. This is our second digestive hormone. Secretin is going to act upon the acinar cells of the pancreas, inducing them to release enzymes. Those enzymes are then going to facilitate digestion of protein, starches, and to a certain extent, perhaps lipids as well. Secretin is also going to act upon the pancreatic ductule cells to get them to release bicarbonate ions. That bicarbonate ion is going to neutralize the acidified material that is now in the anterior intestine. It's going to shut down the action of the acid-active enzymes that were functioning in the stomach, and it's going to allow the functioning of the alkaline enzymes to ramp up. As those alkaline enzymes ramp up and you have additional amino acids, glucose, lipids, and free fatty acids becoming available, that along with the presence of the peptides that were initially produced by pepsin, that's going to induce the I cells of the intestine to produce cholecystokinin. That cholecystokinin will then act upon the gallbladder, inducing it to release bile. The bile is going to facilitate lipid emulsification and digestion, and free fatty acids being absorbed by the intestinal cells. Keep in mind too that cholecystokinin, it's not shown here, but that will also induce the release of additional enzymes. So cholecystokinin isn't just about the gallbladder. That's its primary function, but it's also going to be inducing the production and release of enzymes, especially lipases, that will facilitate lipid digestion and absorption further. So now we have free amino acids, we have glucose and other simple sugars, we have free fatty acids. All of those are going to act upon the K cells. The K cells are going to be releasing something called gastroinhibitory peptide. That will then be acting upon islet cells of the pancreas that will be producing insulin. So the availability of all of these resources, all of these goodies, the free fatty acids, the amino acids, glucose, and other simple sugars, those are going to become available in the bloodstream. That is going to be a trigger for insulin to be produced. Insulin to be produced by the pancreatic cells that will then induce all of those uptake processes to occur in the various cells of the body, and then those anabolic processes to be initiated. So if you understand this figure and the different processes that are involved here, you will do very well on the exam. I know it's complicated, but hopefully there is some value in having this as a study guide. One thing to note here too, GIP at the top, gastroinhibitory peptide, you can see a couple of additional lines coming from that, those dashed lines. Those indicate negative feedbacks. And so this end product, once we have lots and lots of resources made available in the intestine, now we have all these resources available, we're ready to start metabolizing them. It's important at that point to have a negative feedback loop and reduce the production of gastrin reduce the production of hydrochloric acid at that point. That part of the process is complete. We don't need additional gastrin production. We don't need additional acid production. And so GIP helps to shut those processes down or inhibit them. Now, of course, if an additional meal had been consumed, the presence of that meal, that food, would stimulate the G cells and override that negative feedback loop. But I just want you to be aware that if another meal had not been consumed, there would be negative feedback loops that would shut down the early parts of the process, so they were not continuously running and misusing resources. One thing that I think is important to note here is that all of the processes that we've talked about to date, we've been thinking about them in the context of juvenile or adult fish. Fish that have completed their early life history, they've gone through metamorphosis, and they've assumed their adult-like feeding habits. But that is not the case for larval fish. They don't start off that way. There are no herbivorous larval fish. Every single fish starts off as a carnivore. Even if it's going to eventually turn into an herbivore, it will start off as a carnivore feeding on zooplankton. And it will also start off with a relatively poorly equipped, largely non-functional gastrointestinal system. So we're going to talk about the stages of gut development here just to highlight the limitations that larval fish encounter until they have completed metamorphosis and are able to engage in their adult feeding habits. So the first stage is endogenous feeding. This is when the larval fish is utilizing its yolk sac for as a food resource. It is not actively feeding on anything at this point other than that yolk sac. It's during this point that the primordial gut is beginning to develop. So the alimentary canal, the tube, is beginning to develop, but at this point there is no stomach. So even if this is a fish that will eventually develop a true gastric stomach, it doesn't have it yet. So there's no acid production, there is no chemical digestion occurring at this point. What is occurring at this point is largely cell differentiation. So the cells that will become the oxynticopeptic cells of the stomach are beginning to differentiate from these primordial or embryonic cells. The cells that will ultimately become the cells of the pyloric ceca are beginning to develop. So they're differentiating and assuming these different roles, but they're not there yet. And of course, as a result of that, the enzyme suite is not complete. So the fish may be able to produce some digestive enzymes on its own, but it's not producing the complete suite of them. So the pancreas is not producing a huge number of enzymes that will facilitate protein or lipid or carbohydrate digestion. The same is true of the intestine. Those cells are just beginning to differentiate. They're certainly not able to produce all the enzymes that they will ultimately be able to produce. So digestive capacity is very limited at this point. Now, during early exogenous feeding, this is when the larval fish have begun to consume zooplankton. They still don't have a true stomach in most cases. So the structure may be there, but it's probably not producing acid or not producing acid very effectively. So chemical digestion and protein utilization is therefore very limited. What is going on here at this point is largely intracellular digestion. So the fish is going to be consuming a zooplankton. That zooplankton is going to break down in the gut, and whatever digestion processes are going to occur inside the cells of the enterocyte. Or inside the enterocyte cells of the intestine, rather. So those individual enterocytes are going to penocytose or phagocytose material that has broken off from the zooplankton that's been consumed, and they're going to utilize what limited capacity they have to digest that material intracellularly. The other process that's going on here is the use of exogenous enzymes. So when you think of that zooplankton that's now been consumed, it's dead in the intestine or in the stomach of this larval fish. Even in the absence of enzyme production from the fish, that zooplankton still has enzymes that were functional in its own body. Once it's dead, it will actually begin to autodigest, or essentially decay within the intestine of the larval fish. So the own enzymes, the zooplankton's own enzymes will begin to digest its body. And as its body begins to autodigest, It will release proteins, it will release lipids, it will release carbohydrates and break apart in the intestine of the fish, even though the fish has done very little to actually facilitate digestion. Keep in mind that the digestion that's occurring in the fish at this point is not really luminal digestion. It's not producing any acid, it's not producing many enzymes to facilitate attacking that food item in the lumen. It's really only digesting things once it's absorbed them. So once those cells have taken in material from the zooplankton through pinocytosis or phagocytosis, that's when they're able to do a little bit of intracellular digestion. But there is no extracellular digestion to speak of occurring. So their fish is really relying upon the zooplankton to auto-digest itself to liberate some of those resources, and then those resources can be pinocytosed, phagocytosed, and then hopefully further digested and utilized by those enterocytes. But it's a very, very inefficient process. Now later on in exogenous feeding, the stomach will have come up to speed, the cells will be more fully differentiated, so we have a true stomach, a gastric stomach developing. So now this stomach exists as a structure, and it's also producing pepsin, it's producing hydrochloric acid. Now we can really have fully functional chemical digestion occurring. This is also the same time that larval fish will assume its adult feeding habits, or something like its adult feeding habits. So maybe it's going to ultimately be a tuna, maybe it is a tuna, and it's going to be feeding on other large fish as an adult. It might not be feeding on those large fish now, but it's going to be feeding on smaller fish, other larval fish, etc. So it's the size of its prey is not the same as what it will be later on in life, but the type of prey is at this point very similar to what it will be eating throughout the rest of its life. So at this point, enzyme production is at capacity, chemical digestion is highly functional. This is where we have this increase in total digestive capacity, and we have an increase in efficiency as well. Now, of course, the timing of these different processes is going to vary somewhat based on the species. So what we just talked about in the previous slide is a generalized approach, a generalized description of ontogeny of the gastrointestinal system of fish. But again, the timing relative to exogenous feeding and the development of the gastrointestinal system is going to vary. So here we have a description of when spawning occurs, when fertilization occurs, when the eggs hatch, and when they develop and assume their adult feeding habits. So we're actually going to begin in the middle with the agastric fish. So these are the fish that don't develop a stomach later in life. So you can see some examples here. This could be a carp, it could be a goldfish. So at the embryonic stage, so the fish are spawned, the eggs are fertilized, we have a developing embryo, and then we have hatching. That's what's referred to by the H here. So the fish hatches, then we have a free embryo. At this point, it's utilizing its yolk for energy. And then we have first feeding occurring. That's indicated by the F. So first feeding, and then larval development, and then ongoing development. In this case, the fish is not... it's not going to develop a stomach later in life, so the absence of a functional stomach is not a major limitation for these species. What the major limitation is in terms of their ability to fully digest their food at this point is enzyme production. So during those early first feeding stages, they are going to be consuming zooplankton. They are going to be relying upon auto-digestion of the zooplankton in their stomach until their enzyme suite fully comes up to capacity, their microbiome becomes fully colonized with microbes living in their intestine, and their full adult feeding behavior can begin. Now, with respect to the gastric fishes, we can divide these a little bit further into the altricial gastric fishes, shown at the top, and the precocial gastric fishes at the bottom. Let's actually start with the precocial gastric fishes. So in this case, you can see we have spawning, hatching, and then before we have first feeding, we have that G indication. This is when gastric digestion is considered developed. So in the case of the precocial gastric fishes, like salmon and catfish, their ability for, or their capacity for gastric digestion, in other words, the functioning of their stomach and their ability to chemically digest protein, that is developed before they begin first feeding. So these free embryos, these yolk sac fry, have fully developed their stomach before they need to begin first feeding. This is a stark contrast to the altricial gastric fishes that you see at the top, which may be much more familiar to many of us that deal with temperate fishes. Or cool water fishes rather. So in this case, for the altricial gastric fishes, we have the same fertilization, hatching, first feeding, but in this case, gastric development or gastric digestion doesn't occur and fully develop until well after first feeding in these fish. So the altricial gastric is very similar to what we described on the previous slide. These are fish that are going to be very inefficient at utilizing food resources for the first few days, weeks, months of their life because their full enzyme suite is not developed, and their ability to produce acid in their stomach to denature proteins and make them accessible to proteases and peptidases is not functional for a long period of time. So striped bass, halibut, sablefish, lingcod, many other different species are altricial gastric. They have to start feeding exogenously before their gastrointestinal system is fully developed, before they are capable of chemical digestion. Salmon, catfish, and other precocial gastric fishes are not so limited. Their gastrointestinal system is pretty well functional, pretty well developed. They are able to engage in chemical digestion before they ever have to begin feeding off of anything other than their yolk sac. Now, you might be thinking to yourself, why would any species want to have altricial gastric offspring? It would seem to be a much greater advantage to have precocial gastric offspring that don't have to eat exogenously until their gastrointestinal system is fully ready to process exogenous food. And it basically comes down to energy investment. Salmon, catfish, and other precocial gastric fishes have very large eggs, and the females invest a large amount of energetic resources into each one of those individual eggs. And so the number of eggs, progeny that can be produced by the female fish in this case is limited by egg size. In the case of the altricial gastric fishes, these eggs are very small. There's a small yolk sac available, and so the female is able to produce many more eggs per unit of her body mass, but each one of those eggs is invested with less in the form of energetic resources. We'll talk a little bit about these two different types of strategies more when we get to reproduction. Some are better for some type of environments, some are better for others. But in terms of nutritional physiology, the precocial gastric fishes are certainly at an advantage when they begin first feeding relative to the altricial gastric fishes. And this figure just summarizes that, that transition from intracellular to extracellular digestion and how that's going to influence the efficiency of fish in acquiring nutrients and utilizing those resources from their food. So this is a description of an altricial gastric fish. So you can see on the left-hand side we have first feeding. And you see an increase in assimilation. We have total digestive capacity shown in the large hatched line, and then we have extracellular digestion in the short hatched line and intracellular digestion in the heavy solid line. You can see that both there's essentially no extracellular digestion occurring at first feeding. That's because these fish don't yet have a truly functional stomach. They're altricial gastric, they won't develop that functional stomach, and they won't develop the capacity for chemical digestion until much later in development. So that's at zero for a long period of time. The only digestive capacity that they have initially and for a period of time following first feeding is intracellular digestion. So we have a somewhat of an increase to approximately 30% assimilation efficiency, and that's about all these fish are able to acquire. That's all they're able to achieve utilizing those intracellular processes, where the individual enterocytes are penocytosing or phagocytosing material from the zooplankton that they consume and doing the best that they can for intracellular digestion, but it's a very inefficient process. That's why they're never able to achieve better than 30% assimilation when intracellular digestion is functioning. And again, keep in mind that assimilation, this refers to the percent of the food resources that stay in the organism. So at this point, only 30% of the nutrients or 30% of the energy that is present in their food is actually being absorbed by the larval fish. So they're losing 70% of what's theoretically available they're not accessing because they're not able to digest it effectively before the digesta is passed. Now, later on in development, as we lead into metamorphosis, we see an increase in that extracellular digestive capacity. So this is a result of acid production in the stomach becoming fully competent in terms of chemical digestion. We also have an increase in enzyme production. So enzymes that are being produced by the intestine itself, by the liver, by the pancreatic tissues, certainly, all of these that are being released into the lumen of the intestine to facilitate that extracellular digestive process. This is where the real efficiency of digestion comes from, those extracellular processes. So you can see as that process or as that digestive ability increases, we see a dramatic increase in the total digestive capacity. There's always going to be some level of intracellular digestion occurring. We've talked about this already with respect to lipids and proteins. So some of those simple peptides, di or tripeptides, or simple lipid components may be absorbed directly by the enterocyte and then further broken down to individual amino acids or individual fatty acids. Some of that intracellular digestion will continue to occur, but at a relatively low level. As those extracellular processes come up, that's where that efficiency comes from. So we have a dramatic increase in the total digestive capacity, representing largely extracellular processes, but also a little bit of ongoing intracellular processes. The point being that as these fish, these altricial gastric fish, go from first feeding into metamorphosis, we see a dramatic increase in the total assimilation capacity that they have. Early on, these fish are not able to utilize even the best quality food very effectively. Later on, once they complete metamorphosis, they will be able to. In the case of the precocial gastric fishes, their diagram of intracellular and extracellular digestion would look very, very different relative to first feeding. That's the kind of question that I would be very likely to ask you in the exam. Show me a figure of intracellular and extracellular digestion for altricial gastric and precocial gastric fishes, and how that's going to change over time for those two different types of species. Now that we've talked about acquisition of nutrients, digestive processes, absorptive processes, I want to start talking about bioenergetics, which is essentially a study of energy partitioning among anabolic and catabolic processes and the balance between food or energy resources that are consumed and energy that is expended and energy that is retained in the form of new tissues, reproductive products, etc. We oftentimes think about this in the context of aquaculture because bioenergetics will influence proper diet formulation, our ability to formulate a feed so that we have limited waste that's produced in the form of effluents, etc. But of course, bioenergetics is really important for ecological studies as well. A lot of times we think about bioenergetics, or a lot of times ecologists think about bioenergetics in terms of total ecosystems and what is the carrying capacity of that ecosystem and how does energy flow through it. But at the end of the day, all of that boils down to the individual organismal bioenergetics, and the organisms that are present in that ecosystem, of course, are going to drive the overall energy budgets for that ecosystem. So bioenergetics is important for aquaculture, it's important for field biology, it's important for ecology. Bioenergetics models, or the theory of bioenergetics, works based on the first law of thermodynamics, or really thermodynamic theory in general. The first law of thermodynamics is that energy and matter can neither be created nor destroyed. Or to put this differently, what goes in must go out, or it must accumulate. It must pile up in that organism itself. So if we think about this a little bit differently, what piles up, what accumulates in terms of the mass or the energy contained within an organism, cannot be greater than the difference between what went in and what went out. So this is the basic first law of thermodynamics. We're not creating or destroying energy or matter, but we can be changing its form. The second law of thermodynamics also applies here, in that energy can change form, but when we change energy or matter from one form to another, there will always be some loss of energy in the form of heat. And that will come in later when we talk about something called specific dynamic action. So essentially, bioenergetic models are mass balance equations where energy, matter, nutrients are consumed, and those consumed resources are partitioned into their different fates. So energy that is retained in the form of growth, energy that is utilized to support resting metabolism, and energy that is lost in the form of waste or excretions. Now, as we think about these bioenergetic models, there are going to be a number of factors that make bioenergetic models for fish different from those for terrestrial animals. So the first and most important of these factors is poikilothermy. Now, of course, most fish are poikilothermic. This does not apply to the homeothermic fishes like great white sharks and other large-bodied, highly active fishes. We'll talk about them more when we talk about temperature regulation. But for most fishes, they are poikilothermic, and that's going to dramatically affect their bioenergetics. They're not going to be expending energy to maintain their body temperature. And so that means that there's a lot more energy available for other types of processes. Those of you that are involved in aquaculture, this is why fish are able to achieve feed conversion ratios that are substantially more efficient than any kind of terrestrial organism. That energy that a terrestrial organism is consuming, a very large chunk of that is going to be utilized just to maintain body temperature and nothing else. Fish, of course, don't engage in those types of activities, and so they have additional surplus energy, or they're likely to have surplus energy that can be transformed into new tissue growth. The second factor here is reduced gravity. So fish, unlike terrestrial organisms, don't have to invest as much into a very heavy, strong skeletal system to support their weight, because the water around them is going to provide a substantial amount of support instead. So if you think about the bones of a cow or of a dog or of a human, they're much heavier, much stronger, much more heavily mineralized than the bones of an equivalently sized fish. It's not the fish that don't have a skeleton or don't have energy investment tied up there, but it's substantially less than what you would see for an equivalently sized organism living in a terrestrial environment. So that will also influence the amount of energy that they have to put into growth in the form or in terms of energy dedicated to those types of investments or processes. Now, osmoregulatory changes are a little bit different. So when we think about fish that are living in freshwater versus fish that are living in saltwater, they are going to have a certain amount of energy dedicated to maintaining osmoregulatory function and maintaining a proper level of osmolality in their bodily fluids. Now, as we talked about, the energy that's associated with that is going to be influenced by how big of a gradient the fish face. If you're a freshwater fish and you're facing a gradient of maybe 350 milliosmoles per kilogram in your bodily fluids versus approximately 0 in the environment, that's a gradient of approximately 350 milliosmoles per kilogram. That's a pretty substantial difference that you have to maintain, but it's small relative to the gradient that osmoregulating fish, non-osmoconforming fish, have to balance in a saltwater environment. In that case, the osmolality of their fluids is likely between 350 and 400 milliosmoles per kilogram, whereas the environment may be over 1,000 milliosmoles per kilogram. So we're talking about a much larger gradient that those fish are facing. It's a different gradient, it's in the opposite direction, but the difference in magnitude that they're having to maintain in terms of a difference between their tissues, their fluids, and the environment they live in is much larger in saltwater than in freshwater. And so that's going to have different energetic costs to maintain that difference. So whether you're talking about a marine fish or a freshwater fish, that's going to influence their bioenergetic model as well. Of course, bioenergetic models are going to be influenced by the quality of food that the fish is able to acquire. This is true certainly in an aquaculture setting where we have direct control over what the fish are being fed, but it's certainly true in natural systems as well, based on the availability of forage different times of the year or as a result of changes in population structure, introduced species, invasive species, etc. With respect to life stage, we've talked a fair amount about how the gut is not fully developed and the gastrointestinal system is not fully developed during the early stages of larval development, and for the altricial gastric fishes, that will have a dramatic effect on how they're able to acquire and what they're able to utilize from acquired food items. Life stage will also affect fish and their bioenergetic models later on in life in terms of how they partition available resources into growth versus reproduction. So there's lots of different factors here that are going to be affecting bioenergetic models. One thing that you don't have listed here is temperature. Certainly temperature is going to be affecting all of the different elements of bioenergetic models as well. When we think about a bioenergetic model, we can think about it first in the context of a very, very basic energy budget. That's the equation that you see listed here. This is essentially a mass balance equation, and again, it's driven by that first law of thermodynamics, that energy is neither created nor destroyed. So what's on the left-hand side of the equal sign has to equal what's on the right-hand side of the equal sign. So we have C, which refers to the ingested food resources or the ration that the fish are offered. If we're talking about an aquaculture setting, this is the total energetic value or total nutrient value of the food that they consume. That has to equal everything else. Everything else includes production of tissues or other products. So this would include accumulation of new body mass, new muscle tissue, new fat tissue. It also includes other products, so synthesis of new DNA for new cells, synthesis of enzymes, hormones, etc. So it's all the different production of new things the fish is doing with the energy and nutrients that it's consumed. R refers to synthesis and other physiological work. So of course there's going to be some energy expended to produce P, to produce those new tissues and other products. That energy is ultimately going to be lost. A certain amount of that energy is going to be lost in the form of heat. So it still exists, but because we were transforming energy from one form into another, it's going to be lost to the organism. So it's not going to be retained in the form of P or those tissues, but it's still part of the overall equation, even though it's lost to the fish. And then E is also lost to the fish. It's the amount of the ration or the amount of C that is lost as waste products. This is the feces that's produced by the fish. It's the urine that's produced by the fish. It's the gill excretions. It's all of the different elements of matter, energy that the fish consumed but was not able to do anything with and ultimately released, excreted in the form of waste. That's a basic energy budget. Of course, bioenergetic models get much more complicated than that. So again, just an illustration of this with respect to a fish. C is what they're consuming. P is going to be largely associated with their body mass, as well as the additional products like enzymes, hormones, and other things that they may be producing. There's going to be losses in the form of feces, respiration, excretions of other kinds. So C has to equal the sum of all the other different components. If we wanted to get a little bit more detailed here, we could look at each of those elements, P, R, and E, in terms of what they actually entail. So P is going to be the sum of P sub G and P sub R. P might be somatic growth. P sub R could be reproductive components. For a juvenile fish, P sub R is going to be 0, and P is only going to be associated with somatic growth. Later on in life, as that fish enters into reproductive maturity, P sub R is likely to become dominant, whereas P sub G is going to decrease dramatically and may actually become 0 for adult fishes, for some adult fishes. R is going to be total metabolism, total respiratory costs. These are the losses for a fish, that energy that's expended and cannot be retained. R is a lot more complicated. So R includes R sub S. This is the standard metabolism. This is the fish at rest. This is how much energy a fish expends just existing. R sub R is routine metabolism. So this is how much energy a routinely active fish is going to be expending. So this isn't a fish just sitting there. This is a fish engaging in its usual day-to-day activities: swimming, avoiding predation, acquiring food, etc. Whatever it actively typically does, that's the routine metabolism. Then you have feeding metabolism, R sub F. This is also referred to as specific dynamic action. This is the amount of energy that's associated with metabolism of resources. So, for example, we can take glucose that's absorbed by a hepatocyte and we can transform that glucose into glycogen for storage. We can transform glucose into glycogen, but as we transform that energy or that nutrient from one form to another, some of that energy is going to be lost in the form of heat. That heat that's lost is specific dynamic action, transactional costs as we transform one form of energy into another. So that's a part of this as well. R sub A is active metabolism. This is going to be fish that are doing a little bit more than routine metabolism. So this could be a fish and its metabolism, its energy expenditure during a migration, for example. It's not part of its routine daily activity, but it's something that a fish is engaging in periodically throughout its life cycle. So a salmon that's returning to its natal stream would have a much higher active metabolism that would be beyond its standard metabolism of feeding and other behavior when it's in the ocean. Each of these components is then going to be multiplied by a factor that represents the percent of time spent at each activity. So throughout the day, a fish is going to spend a certain amount of time at rest. So maybe 10% of its time it's at rest. So 10% of 10% would be multiplied by that metabolic rate R sub S. Maybe it spends 90% of its time during certain seasons engaging in routinely active behavior. So routine metabolism, in that case, R sub R would be multiplied by 90%. In the case of that fish, that salmon that's returning to its natal streams, maybe R sub A, active metabolism, represents 50% of its activity at that time. Then the rest of the multipliers for the other factors would have to change accordingly. So those multipliers represent the percent or the fraction of time that's spent engaged in any activity. Again, you can see where just one component of this simple energy budget, so-called simple energy budget, just one component, R, is actually representative of many other different components, which are, again, it gets much more complicated as we get more detailed and more accurate in our terms of our description of an energy budget for an organism. The same is true for E, or the excretory products, the waste products. That's going to be representative of feces as well as other excretions like nitrogenous waste that's excreted at the gill, so ammonia or urea for those ureotelic fishes that we discussed before. So even this still fairly simple equation or energy budget, if we put it all together, it would be much more complicated than that initial C equals P plus R plus E would imply. Again, this is still a pretty simple energy budget. If we wanted to get more detailed and more accurate, each of those components could be further subdivided into their constituents. So it gets pretty complicated pretty quickly. This is a slightly different way to look at an energy budget or a bioenergetic model that I think might be a little bit easier to understand. So what's shown in green, these are the positive energy fluxes. So this is essentially what's coming in different forms. What's shown in red, these are various types of losses, and then what's shown in yellow is retained energy or energy that the fish can actually utilize. So let's assume the top that we have an intake of energy of 100 calories. So a fish consumes a crustacean, a crab, let's say, and that crab has 100 calories of energy associated with it. Now there's a certain amount of that crab that might not be digested. Let's say that the crab has a lot of chitin in its exoskeleton and the fish does not produce enough chitinase on its own, and its microbial community, its microbiome, is not able to produce enough chitinase either, and so a certain amount of that chitin is going to pass through the fish undigested. So we may lose 4 calories of energy in the form of fecal energy. So this is 4 calories that the fish theoretically could have utilized but was unable to utilize because they weren't able to digest that chitin. So in this case, now we're down to 96 calories of digestible energy. So this is what the fish was actually able to digest and acquire. Now then we have urine energy, branchial energy. These are additional excretions, additional losses. So they're similar to feces, but this is the amount of energy that theoretically would have been valuable to the fish, but the fish lost in the form of urine excretions and excretion of nitrogenous waste at the gill. So we may lose an additional 11 calories there. What's left over then in green is metabolizable energy. So this is the energy or the nutrients that the fish are able to potentially do something with. But you can see there's a final loss here. This is called heat increment. This is the same thing as specific dynamic action. These are those transactional costs that we discussed before in terms of what the fish is doing with these nutrients. And in making some of those transformations, they're going to lose some of the available energy in the form of heat. Let's say an additional 5 calories. Then what you're left with is net energy. So this is what the fish is able to utilize. They've had all these other losses, and now they've got what's left. So the net energy is then going to be divided between basal metabolism, voluntary activity, and recovered energy. So basal metabolism, this is similar to what we thought of as a resting metabolism or a routinely active fish. This is, again, what the fish is typically doing on a day-to-day basis and how much energy they have to expend to do that. Then we have voluntary activity. This again could be the maybe a voluntary isn't such a great way to put it. This is the activity that goes above and beyond the routine activity of a day-to-day basis. So this could be activity associated with a migration, for example. Then you have recovered energy. This is what the fish can actually retain within its body. So basal metabolism, voluntary activity, those are energy expenditures that the fish has to make, but ultimately you can't see it. It's lost to the fish. They expend that energy and then it's gone. Recovered energy is what will accumulate in the form of their body. So recovered energy could include somatic growth. It could include egg or milk production and a variety of other different things. Another way that I like to think about these bioenergetic models is in the context of not an energy budget, but a financial budget. So at the very top, the intake of energy, this could be your gross salary, the total amount of money that you are able to acquire on a monthly basis. That's your monthly salary up at the very top. Now, there's a certain amount of money that's going to be held back from your paycheck that's going to be used for other purposes. So it might be for insurance, it might be for Social Security, it might be for health care. So there's a certain amount of money that's held back from your gross payment that you never really see in terms of your paycheck. That's going to be held back. That's something that might be equivalent to digestible energy. So we've gone from our gross salary to our net salary. So that's equivalent in this case of going from our intake of energy to our digestible energy. So now we've got our net paycheck, our net salary, and it's in the form of a check from our employer. But you can't go to the grocery store with a check from your employer. You have to transform that check into another form of currency. So you might take it to the bank, but they'll change it for you. They'll turn it from a check into cash, but it's likely that they're going to charge you a fee for making that transaction. That's equivalent to heat increment. So we're taking our energy, our currency, our salary, our net payment, and we're transforming it from one form into another form, but we're going to lose a little something in that transaction. So we're going to pay a transactional cost, we're going to pay a fee to the bank for transforming our paycheck, our net salary, from a check from our employer into cash that we can actually spend. That's equivalent to what these fish are doing when they have metabolizable energy relative to net energy. They're transforming the energy from one form into another, and they're going to have to pay some transactional cost in the form of energy that's lost as heat. So that's that heat increment or specific dynamic action. Then you actually have your cash money in your hand. Now you can decide what you need to do with it. Well, there are certain bills that you simply have to pay. You have to pay your rent. You have to pay your utility bill, at least in terms of if you still want to have a place to live with power and water serving it. So there are certain things that you have to do with that money that you don't necessarily like to do, but you have to do. That's what would be an equivalent. That would be equivalent to basal metabolism or voluntary activity. The fish might not want to engage in predator evasion or acquiring food, or they may not want to expend energy for migration, but they have to. Then what's left over after that, after you've paid all of your bills, you've paid your rent, you've paid your utilities, what's left is what you can use for fun. You might be able to use that money to go to the movies, to buy a 6-pack of beer, to buy a jet ski. things that you would like to have or like to do, but don't necessarily have to have or have to do. In that case, for fish, that's recovered energy. They've paid their utilities, they've paid their rent in the form of basal metabolism and voluntary activity. Now they have what's left over for them. They have what's left over for fun. Now, in the case of fish, they don't want to buy a jet ski, but what they do want to invest in is somatic growth or reproduction. So again, I think it's useful to think of these energy budgets in a very similar manner to financial budgets. It's the same sort of thing. There's the gross amount of money that is available, or the gross amount of energy. Then there's what's actually digestible, what you get in your paycheck. Then there's what's available to you after you've paid all of your transactional fees, and then what's available for fun and activities that you want to do and activities that you need to do. It's the same thing for fish. There's the total amount of energy that's available, there's the digestible energy, metabolizable energy, and then once they have their net energy, they can make decisions as far as what they need to do with that based on what they have to do, energy demands that they have to fulfill, and then those that are optional but certainly desirable. So when we talk about what the fish might do with investments of energy, with its net energy that's left over, hopefully the fish has something left over after it's paid its rent, after it's paid its utilities in the form of basic resting and active metabolism. Hopefully it has enough energy left over to invest in growth. So what exactly is growth? Well, it depends on who you ask. There's lots of different ways to think about growth. You can think about growth as being an increase in cell number or an increase in body size. But growth also includes things like redistribution of resources. So, for example, if you have a fish that is becoming reproductively mature, like a pink salmon. A male pink salmon, these are often, the species are referred to as humpies because the males develop a very large hump on their back that's used for digging redds in the gravel in which these, in the streams in which these fish spawn. They also develop hooked jaws, large teeth for defending their potential nest areas, their potential redds from other competing males, and again looking attractive to females. That is an aspect of growth as well. They are redistributing resources. They may not be changing their body mass at all, but they're certainly changing their morphology, taking energetic resources that may have been held in the form of muscle within their trunk or lipid that was stored in their gut, and they're redistributing that to form that hump-like structure. They're redistributing that to form these long teeth to make them more successful in reproduction. That's not what we typically think of as growth, but that is one aspect of growth and resource investment that fish engage in as well. Growth also includes, or investment of resources and growth includes something called tissue turnover, or what I refer to as the Red Queen paradigm. In Alice in Wonderland, the Red Queen was running and running and running just to stay in place. Every organism is continually engaging in tissue turnover. They are investing energetic resources and investing nutrients in replacement of tissues, products, enzymes, hormones, things that get used up and go bad. So as one cell begins to degrade and ultimately dies, it has to be replaced with another. Again, we don't typically think of that as growth, but that's an element of growth. It's investment of available resources and assimilation of those resources into the body. So those are different aspects of growth, and certainly how you define growth will vary based on who you're asking. If you ask a fish farmer or a regional biologist, a fisheries manager, growth to them would likely represent changes in length or weight or condition of fish, increases in the size of the population. It might include gonatosomatic index. It might include dress out, or the amount of edible product that you can harvest from a fish. If you were asking a physiologist, they take a very different perspective. They might think of growth in terms of a change in the RNA to DNA ratio. If you have increasing amounts of RNA, that suggests those cells are producing more proteins. So as the RNA to DNA ratio increases, that means you have more RNA being produced, and of course the same amount of DNA. That would suggest that those are growing active cells. You might also think about growth in the context of nitrogen retention. How much nitrogen is going into the fish and how much is staying behind. You might think of it in terms of amino acid uptake or retention in terms of new protein synthesis. So growth and assimilation of resources can reflect a lot of different elements. And so I think it's important to just recognize those and understand that growth isn't just an increase in total length or an increase in biomass. Typically, we think of fish as having a growth curve that's sigmoidal and looks something like this. So as fish age, they increase in size fairly dramatically during juvenile and early adult stages, but ultimately that growth is going to slow down as they become larger and larger, or at least growth in the context of body weight is going to slow down. They're going to reach a relatively, not maybe a true maximum size, but certainly a size at which they're not likely to get much larger. This is because the bioenergetic model at those later ages is going to shift. During the early stages, the fish are focused on somatic growth because there's an evolutionary drive to become bigger. When you're bigger, there are more things that you can eat and fewer things that can eat you. So there's a significant advantage to acquiring larger body size relatively quickly during those early stages. So any energy that's available that's surplus to those basic metabolic demands for a young fish is going to go directly to somatic growth. Now for other fish, older fish that have achieved a certain size, at that point their bioenergetic model is going to shift so that any surplus energy that they have beyond their basic metabolic demands, it may be more attractive, it may be more advantageous for them to invest that available energy and invest those surplus resources into reproduction. So if there's more energy being devoted to reproduction, that means there's less energy available for further somatic growth, and so you tend to see this sloping off of growth as the fish age. So we commonly think of fish as being indeterminate growers. In other words, they can continue to grow throughout their lifespan, but this is something that's not entirely true. It's true that many fish may continue to grow a certain amount as they age, but they're all going to reach that maximum or that threshold where most of the energy available is going to be dedicated to reproduction. They're not going to be investing much in the form of somatic growth, and so they will reach maybe not a absolute maximum size, but certainly something approaching that. Now, there are certain fish that are much more like a determinate grower. Zebrafish, for example, never really reach a size beyond maybe an inch. Many of the aquarium fish that we commonly have in our homes are much more like a determinate grower, like humans or many terrestrial animals. Humans don't continue to grow throughout our lives. We reach a certain size and then we stop growing. Our energetic models then shift to invest energetic resources into other things. Many fish are much more like that. Again, I gave the example of zebrafish. They might technically get a little bit larger as they grow as they get older, but not substantially. So when people tell you that fish are indeterminate growers, for many species of fish, that's simply not true. They do reach a maximum size and then do not grow beyond that. For many other fishes, it might be technically true that they are indeterminate growers, but in a practical sense, they do tend to reach a near maximum size and not grow much beyond that. So that's the distinction between determinate and indeterminate growers. And another thing that's commonly said about fish growth is that it is continuous. But growth cannot be continuous in fish. It looks continuous. It looks like it's a continuous process, so the fish just increase in size smoothly. But if you look closely at that curve, it's much more of a discrete process. There may be days or hours or seasons or weeks where you have an increase in body mass, but then there are also decreases in body mass, or times where growth, at least in terms of body weight, is static and does not change. So when people say that growth is continuous in fish, that's not really true. Growth is not continuous because the acquisition of resources is not continuous. If fish were continuously eating and continuously acquiring resources on a minute-by-minute, second-by-second basis, well then yes, growth could theoretically be continuous. But the fact is that feeding behavior and feeding events and intake of energy occurs in discrete moments in time. And growth can only occur after those events occur. So the overall process may appear to be a smooth, continuous process along that sigmoidal curve, but on a daily basis, on an hourly basis, when you zoom in more closely, it's a much more discrete process reflecting individual feeding events. Now, the growth rate of fish, there is a theoretical maximum growth rate for all fish that's going to be influenced by their ability to acquire food or the feeding rate that's offered to them in an experimental or an aquaculture setting. It's also going to be influenced by a host of other different things, including water temperature and life stage, etc. But of course, their ability to acquire food is going to be a pretty dominant factor in that. Here you can see the theoretical growth rate for a variety of different fish. So we've got a yellow fish, a green fish, and a pink fish. Each of these curves shows the growth rate in terms of percent body weight per day or any number of other different factors, but the accumulation of body mass per unit time as a result of different rations or the amount of food that the fish acquires or is offered every day. Well, you can see that there's a certain ration for each of these fish that is necessary just to achieve a 0 growth rate. So just to maintain the size of the fish, just to maintain their body weight, there's a certain ration that is needed. The pink fish requires a little bit more food on a daily basis than the green fish does as a maintenance ration, just to maintain their body size. In this case, the pink fish might represent a larval or early juvenile fish, whereas the green might represent the same species of fish at a later stage in its life. Small fish have a very high resting metabolic rate, so they need more energy per unit body mass just to maintain their size. So to achieve 0 growth, to maintain their body mass, they have to consume a greater ration, they have to consume a greater percentage of their body weight every day relative to an older individual of the same species. It's important to note here too that if they're consuming less than that, their growth rate will be negative. The fish during starvation can decrease in size. It's not that they will necessarily decrease in their length, of course, but their body mass certainly will decline if they are not able to achieve at least a maintenance ration for a significant period of time. Now, of course, as you get beyond that and you increase that ration further, the growth rate will increase correspondingly up to a point, to G max. Now, what G max represents for each of these individual fish or these different species of fish, whatever these lines represent, will be influenced by life stage, water temperature, and composition of the ration. How much protein is there? How much fat is there? This is something that's important to understand, that our optimal and G max, the maximum growth rate and the optimal amount of food that's consumed by a fish on a daily basis will vary with those life stages, with those environmental conditions, with water quality, temperature, etc. So what's achievable in an aquaculture setting, we can probably manipulate the environment and the ration and the quality of the food to achieve those maximum growth rates. It's very unlikely that fish are able to achieve those conditions in most environments in the wild. They're certainly not able to achieve that in most cases throughout the year. So the growth rate for a fish, it could be the exact same fish, green, yellow, or pink line, but just at different seasons throughout the year or when different forage or prey is available. So understand that there is a maximum for any set of conditions for any species, but that is going to be influenced by the environment as well as food acquisition. So of course, investment in growth, whether it's somatic growth, tissue turnover, reproductive investment, these different investments will be indicated based on different exogenous factors. So we've talked about how temperature affects everything in fish. Temperature and the availability of exogenous food resources, this is going to influence tissue turnover rates. It's going to influence those basic metabolic requirements. So that will alter those bioenergetic models and determine how much surplus energy is really available for things beyond basic metabolism. And that will, of course, influence how those available surplus resources get invested. Temporal factors will also influence growth. Temporal factors including seasonality, although one could argue that seasonality is really just another way to describe availability of food resources and water temperature, those other exogenous factors that we've already talked about. But temporal factors can also include age of the fish, whether or not we're talking about a juvenile fish, an adult fish, a fish that's engaging in reproductive development, etc. But of course, these exogenous factors, including aspects of the environment, aspects of the age of the fish, the time of year, those are not going to influence growth directly. Rather, they're going to be part of the information that influences growth. But all of that is going to be synthesized through endogenous factors. So growth and all the different aspects of growth are going to be controlled by the endocrine system as well. The endocrine system is going to be integrating those exogenous factors. The endocrine system of the fish and hormone levels are going to reflect water quality. They're going to reflect time of year. They're going to reflect age of the fish. They're going to reflect available energetic resources. But the hormone system, the endocrine system of fish, is also going to reflect other endogenous factors. So how many amino acids are available? What types of amino acids are available? How old is this fish? How many times has it reproduced? These other different types of internal decision-making processes are going to be influencing the overall endocrine state of the fish and how available resources get invested. So it's going to be internal factors, external factors, and as we'll discuss later on in the course, the endocrine system is going to integrate all of that and ultimately help the individual cells and the organism as a whole make decisions about how available resources are ultimately invested.